Hello everyone, so this is the GCSE Combined Science Trilogy paper walkthrough for Biology Paper 1, and this is the Foundation Tier paper. This was the paper that was sat by pupils in May 2018. Now I've attached beneath this video the actual link to the paper and the mark scheme, so you can go through that as well. I am going to model how I would have tackled the paper, question by question, and I've also included revision video links to help you revise the specific topics for each question if you struggle on it. So starting now then, I will go through the paper. So question number 1, figure 1 shows a type of white blood cell. Figure 1, and you've got a label pointing to A. What is structure A? Tick one box. So I should think to myself, what type of cell is a white blood cell? I should remember, well, it's a type of animal cell. So therefore, it can't be a cell wall because that applies to bacteria or plant cells only. And because it's on the outside of the cell, it's not going to be a cytoplasm or a nucleus because they are on the inside of the cell. Therefore, it's going to be a cell membrane. Question 1.2, white blood cells help to defend the body against pathogens. How do white blood cells do this? Tick 3 boxes. Well, you've got a whole list here and you know you've got to tick 3 of them. So I would go through each one in turn to begin with and then identify which ones you're going to put as your final answer. So the first one is cloning pathogens. Well, our white blood cells wouldn't want to make more pathogens, so we don't think it's going to be that one. Engulf pathogens, well, that sounds likely because they're going to swallow them up and digest them. Produce antibiotics, well, antibiotics, they're a drug that a doctor provides to kill bacteria. So our white blood cells aren't going to make antibiotics, but they might, and they do, produce antibodies. Finally, you've got the options of producing antitoxins or toxins. Well, your white blood cells are there to defend you, so they're not going to be making toxins, so it's very much they produce the antitoxins to neutralize the toxins. Measles is a serious disease. A person can die from measles. Figure 2 shows the number of cases of measles in England and Wales between 2012 and 2015. Use figure 2 to calculate the decrease in the number of cases between 2012 and 2015. So if we look at the graph, we've got the bars for 2012 and 2015, and we need to calculate the decrease in the number of cases. If we look at our y-axis then, we can see it's going up in 500s. For every 10 little squares, that is equal to 500 cases. So therefore, one little square is therefore going to be equal to 50 cases. So if I look at 2012 to begin with, I can see reading off my graph is one little square above 2000, so that tells me there's going to be 2050 cases. If I look at 2015, it's just 2 little squares up, so it's going to be 100 cases. Now, if you got this far, that would get you 1 mark. But we want to calculate the decrease in it, so you would do 2050 minus 100, which gives you 1950 cases. Question 1.4 suggests one reason for the decrease in the number of cases between 2012 and 2015. Well, there could be numerous reasons for why measles has decreased, but a key obvious one might be if there are more people are vaccinated, okay, which would then stop the disease spreading. Question 1.5. Antibiotics cannot be used to treat measles. Suggest why. So, I would think about everything I know about antibiotics. Well, antibiotics, I know, kill bacteria. We're being asked about measles. Even if you're not sure what causes measles, you know antibiotics kill bacteria and they can't be used to kill or treat measles. So therefore it tells you measles is not a bacteria. So you could say measles is not a bacteria, okay, and therefore it is a virus. Question 1.6. Gonorrhea is a disease caused by a bacterium. It can be treated with antibiotics. Give another way to control the spread of gonorrhea. Okay, so you should know that gonorrhea is a sexually transmitted infection. So therefore, it's spread through sex. It can be killed by antibiotics, but we're on another way of controlling the spread. Well, the easiest way of controlling or stopping STDs from spreading is wearing protection. So you could put protection, or be specific about the type of protection, maybe a condom. You've then got a scientist investigated how effective different antibiotics were at killing gonorrhea bacteria. This is the method that they used. They grew bacteria on an agar in a petri dish. They placed one paper disc soaked in water onto the agar. They placed 4 other paper discs, each soaked in different antibiotics, A, B, C, and D, onto the agar. They used the same size paper discs and the same concentration of each antibiotic, and they put the petri dish in it for 3 days, or they incubated the petri dish for 3 days. So figure 3 shows the scientist's results. A clear area around the disc means the antibiotic has killed the bacteria. So I can see that just by looking at it, I've got my paper discs here, A, B, C, and D, and the one soaked in water, and you've got these areas around each one, and this is where the bacteria has been killed, and where it is grey is where the bacteria is still growing. Okay, so even before I've looked at the questions, they've given me this whole practical, so I could think about my variables. The independent variable is the one thing that you change in an investigation. So I can see that what they are changing is the types of antibiotics. What they are measuring is our dependent variable, where they're measuring the area in which the bacteria is killed. And then they have given us some details in the method of things they've done to keep it the same, or keep control variables the same, because you only ever change one thing. So things they have controlled is the same paper discs, same size of them, and the same concentration of each antibiotic, and they incubated all of them for 3 days. So these are the control variables, and the reasons we use control variables is to ensure we get a really accurate result. Then, if I actually look at the questions, question 1.7, give one control variable the scientists used. Well, we've just already thought and looked about it. So we could say the same size as of paper discs. Or any of the things that we've just said. Suggest why one disc was soaked in water. So water is obviously not an antibiotic. It's just been put in there to compare. Okay, so you could put it's to see a difference with no antibiotics. Okay, we call this sort of experiment a control experiment. Question 1.9. Which antibiotic in figure 3 would be the best to use to treat gonorrhea? Give a reason for your answer. Well, if we have a look at them, we can see just by visually inspecting that A has the biggest area around it where the bacteria has been killed. So we could say it is A, the reason why it has the biggest area where bacteria have been killed. So therefore, it's the best at treating gonorrhea. Okay, so moving on to question number 2. This question is about photosynthesis. What are the two products of photosynthesis? So products means what you get out of a reaction. If we think back to our photosynthesis equation, you should know that it's carbon dioxide that reacts with water, which makes glucose, which is C6H12O6, plus oxygen. So we are after the two products of the reaction, which is glucose and oxygen. A student investigated the effect of light intensity on the rate of photosynthesis, and they had this equipment. They've got the LED light source and they've got pondweed in sodium hydrogen carbonate solution. This is the method that they used. They placed the pondweed 5 centimeters from the light source. They measured the rate of photosynthesis by counting the amount of bubbles produced in 30 seconds. And they repeated the investigation with the pondweed at different distances from the light source. So the question is, how could the students measure the rate of photosynthesis more accurately? And we've got to tick 2 boxes. So the first option is count the number of bubbles produced in 1 minute. If we remember, the students in their method did it in 30 seconds. If we increase the time to a minute, that would decrease the effect of any miscounting in the overall results. So doing it over 1 minute does make it more accurate. Measure the change in the mass of pondweed in 30 seconds. Well, it's unlikely that the mass of pondweed is going to change in such a short length of time, so it's not that one. The third one, measure the volume of gas produced in 30 seconds. Well, if you're actually measuring the volume of gas, that's a much better way of getting a precise value compared to counting bubbles, because you can miscount bubbles, or you could count the same bubble twice. So therefore, that does improve the accuracy. If we just read the other two just to make sure, place the pondweed further from the light source. Well, that would affect the rate of photosynthesis, but wouldn't help us to measure it more accurately. And again, use water instead of sodium hydrogen carbonate solution. That would affect the rate of photosynthesis, but again, it wouldn't help the method measure it more accurately. So it's not those two. Question 2.3. The LED light source does not get hot. Why is that important? Well, temperature is one of the factors that affects the rate of photosynthesis. If it gets hot, it's going to change the amount of photosynthesis happening in the pondweed. So why is it important then? Because temperature affects the rate of photosynthesis. So therefore, keeping the temperature the same is a control variable. So, table 1 shows the students' results. You've got the distance of the light source from the pondweed in centimetres going down, and then you've got the number of bubbles produced in 30 seconds. So you can see the pattern before you even look at the questions. As distance is increasing, the number of bubbles is going down. If we relate that to light intensity, obviously you have more light intensity when it's closer compared to when it's further away. So you know that when there is more light, there is more bubbles, there's more photosynthesis, and when there is less light, there is less bubbles and less photosynthesis. Calculate the number of bubbles produced in 2 minutes when the light source was 10 centimetres from the pondweed. It's a 1 mark question, and because they've just given us this table, we of course need to go back to this table to use it. So it asked us how many bubbles were made in 2 minutes, and this table is telling us how many bubbles are made in 30 seconds. And we're specifically looking when it's 10 centimetres from the pondweed. So 10 centimetres in 30 seconds makes 13 bubbles. So therefore, we have 120 seconds, okay, and in 30 seconds, it makes 13. So if you times 30 by 4, that gets you to 120. So if you times 13 by 4, okay, 13, 26, 39, 52 is the amount of bubbles that would be made in 2 minutes. It then asks you to plot the data from table 1 on figure 5 and to draw a line of best fit. I've already pre-plotted these points on here, and you would get 2 marks for accurate plotting. You would then get 1 mark for your line of best fit. So, assuming that you've plotted your points correctly, you could then, and I always find it easier to turn the page at this point, is draw your line of best fit, and it should be a nice smooth curve going round. Remember, in science, your lines of best fit can either be curves or straight lines. Normally in maths papers, it's normally always a straight line, but in science, they can be curved as well. Question 2.6, give one conclusion that can be made from these results. Well, we can see as the light distance increases, the number of bubbles decreases. So you could say, as distance increases, the amount of photosynthesis decreases. You can also link it back to light as well. So as light amount decreases, the amount of photosynthesis decreases. Or any statement in reverse that means the same thing would get you the mark as well. So you could say the more light there is, the faster the rate of photosynthesis. Okay, so moving on to question number 3 then. A student investigated the effect of different concentrations of sugar solution on pieces of potato. So this is the required practical for osmosis that hopefully you would have done in school. So this is the method that the students used. They cut 3 pieces of potato to the same length, they dried each piece on a paper towel, and they weighed it. They place each piece into a different concentration of sugar solution, and they leave all 3 pieces in there for 2 hours. They removed the 3 pieces of potato from the solutions, and they dried each of them on a paper towel. They then measured the length and mass of each potato after the 2 hours. So figure 6 shows how the investigation was set up. So you've got tube A, B and C, and you can see that tube A was pure water. There was nothing else dissolved into it, so that would be pure distilled water. Tube B has some sugar in it at 0.4 moles per decimetre cubed. You don't need to worry at this point what moles per decimetre cubed means. You just need to be aware that there's some sugar in it. And you can see test tube C has much more sugar in it, 0.8 moles per decimetre cubed. Question 3.1 says, why did the student dry each piece of potato before weighing it? Well, if there was already any solution or water on the outside of the potato, then that would increase the mass. So any excess water would increase the mass. In this practical, we are after how much the potato weighs and what is inside the potato. We're not interested on what is on the surface of it. Okay, so why do we need to dry each piece? So it's to remove any excess water because it would increase the mass. Question 3.2, what two changes would you expect to happen in potato tube A after 2 hours? So we know that tube A is our pure water. Okay, so you've got a piece of potato in the cylinder that is surrounded by pure water. Now, if we think back to our definition of osmosis, osmosis is when water moves from high concentration to low concentration. Now, if I've got pure water on the outside, that means there's going to be 100% pure water on the outside, and there would therefore be less water inside the potato. So therefore, the water is going to move inside. Now, if I've got water flowing into my potato, we can think about what's going to happen to it. Would it break into pieces? No, just because water's moving into it doesn't mean it will break into smaller pieces. Decrease in hardness? Well, let's see what other options there are first. Decrease in size? Well, it's not going to be that because we've got water flowing into it. So it would increase in mass and increase in length. Now, some people do often put decrease in hardness because they think, well, it's in water for a long period of time, but we're focusing on the effect of osmosis. Okay, we know water's moving into it, so its mass and length are going to get higher and bigger. Question 3.3, complete the sentences. Water moves into and out of cells by a process called osmosis. Water would move blank the potato cells in tube A. Well, we know it's going from high to low, so it's moving into the potato cells in tube A. The solution outside the potato in tube A is at a blank concentration than the solution inside the potatoes. So we know because it is pure water, there is no sugar in it, therefore it is at a lower concentration of sugar compared to what would be inside the potato. Question 3.4, the potato in tube B did not change. Give one conclusion that could be made about this observation. So if you've got a piece of potato in your water and you leave it for a long period of time and there's no change of mass, that must mean you've got the same amount of water coming out of it as is going into it. So that is telling you the concentration of the potato, of the solution in the potato, is the same concentration as what's on the outside. So you could summarize it by saying the concentration of the sugar solution is the same as the solution in the potato tube. Okay, so and we know that from, if we go back and look at our test tube B, that is 0.4 moles per decimeter cubed. Okay, but the key thing is you get the mark for saying, for realizing that the concentration is the same. Question 3.5 says figure 7 shows the root of a germinating seed. Describe 2 ways the root is adapted to absorb water efficiently. Okay, so we've got a seed here and we can see the roots coming off it. And we're basically asked how are the roots adapted to efficiently absorb water? Well, we can see in this picture that the roots have an awful lot of root hairs. Okay, there are root hair cells that are adapted to absorb more and more water because they give it a big surface area that can reach into the soil and absorb more. They also have, that you could add, is they have really thin walls to absorb more and more water quickly. So your key things for adaptations is that they have root hair cells that gives it a big surface area, and they also have thin walls. Okay, so moving on to question number 4. Exercise can improve health. A student measured her breathing rate at rest, when walking, and when jogging. Figure 8 shows her results. So if we take a moment just to look at this graph carefully, you've got at rest, walking and jogging, and you've got the breathing rates in breaths per minute. Compare the breathing rates when doing these 3 different activities. Use figures from figure 8 in your answer. So the first thing that I would do, if it's specifically telling me to use values, make sure we actually have our values ready. Read the scale for each one. So the breathing rate at rest, you can see, is 12, walking is 24, and then jogging is 60. So we need to make sure that we've got these numbers in our answer somewhere. And we can look at the differences and compare these 2 numbers. So if we start off then comparing rest and walking, it's 12 to 24. Well, we know that 12 goes into 24 2 times, so therefore we can say the breathing rate when walking is twice or 2 times than at rest. Okay, so we've compared rest with walking, and then we can compare rest with jogging. Okay, so now I can work out how many times 12 goes into 60, which is 5. So we can say, as point number 2, our breathing rate when running, or jogging, sorry, is 5 times than at rest. Okay, then finally, for our third mark, we could compare walking and jogging. So it's how does 24 compare to 60? Well, if you double 24, you get 48, and then it's another 12, so it's half the amount of 24, so it's 2.5 times bigger. So your final mark would be the breathing rate when jogging compared to walking is 2.5 times bigger. Okay, and you could also quote these specific numbers in your answer as well. Okay, but that would get you your 3 marks. When you get about halfway through the foundation paper, you will start realising and seeing that more questions are slightly more extended writing rather than as many 1 and 2 mark questions. So question 4.2 then. Explain why the breathing rate changes when doing different activities. So why does our breathing rate increase when we are jogging? Why is it much higher compared to when we are at rest? Well, we can break it down into our 3 different marks. So we got to know we need 3 short bullet point sentences. So when exercising, we need more oxygen to our muscles. Why does our muscles need more oxygen? Well, it's because they are doing more respiration. Which is aerobic respiration because they're using the oxygen. And what does that respiration give us? It's to provide us with more energy. So you can summarise that question in 3 bullet points there. Okay. Then question 4.3, the heart is a double pump. You've got this figure here, figure 9 here to help you. Describe what this means. So if it is a double pump, like we have in our circulatory system, some organisms have a single pump system, like fish, so their blood just goes around in one loop, but we've got 2 loops. That's because we have a right side of the body and we have a left side of the body, right side of the heart and a left side of the heart. So describe what this means. The right side of the heart, its job is to transport blood to the lungs. That's when it's already gone round the body once, it's used up its oxygen that it had, and it needs to get new oxygen and release and get rid of any carbon dioxide it's picked up. And then it delivers that oxygenated blood to the left side of the heart, which then pumps it round the body again. So the left side of the heart, its job is to transport oxygenated blood to the body. So the right side of the heart pumps it to the lungs to get the oxygen, the left side of the heart then pumps that oxygenated blood around the body. So it's a double pump system. Question 4.4. The wall of the left ventricle is often much thicker than that of the right ventricle. Suggest one reason for this. Well, as we've just said, the left ventricle has to pump blood around the whole body. The right ventricle only has to pump blood to the lungs. So in terms of distance, the left side of the heart has to pump blood much, much further. So the left side of the heart has to pump blood much further. And that's enough because it's only a 1 mark question. Okay. Then question 4.5, people are encouraged to exercise after recovering from a heart attack. Suggest a reason why. If the command word in the question is suggest, it just means put down a logical reason why. Okay. So why might people be told to exercise after recovering from a heart attack? Well, it could be because they need to strengthen their heart muscle. It could obviously then reduce the chance of another heart attack. And then finally, it could be because it also reduces the chance of any fatty deposits being deposited on the walls of the arteries, which could then cause another heart attack. But you just need one of those reasons, okay, to get the 1 mark. Okay, so question number 5. Cells divide in a series of stages called the cell cycle. Stage 2 of the cycle is mitosis. Figure 10 shows a simplified cell cycle for the human body. So you've got stage 1, stage 2, which is mitosis, and then stage 3. Draw one line from each stage in the cell cycle to what happens during that stage. So you've got stages 1, 2, and 3, and then what is happening. You should think about what you know about mitosis, okay, and remember it's one cell splitting into 2 copies that are genetically identical to the original cell. So it's making 2 daughter cells from the one original cell. Now, in order for these cells to be genetically identical, the first thing that needs to happen is that copies of DNA are then made. Once those copies are then made, that allows the nucleus to divide, which is stage 2, and then stage 3, the cytoplasm, the cell membrane can split to make those two genetically identical cells. The mass of the DNA in the human body at the start of the cell cycle is 6 picograms. What mass of DNA will be in each of the new cells produced by this cell division? Tick one box. So as we just said, mitosis gives genetically identical cells. So if you have 6 picograms at the beginning, you will have 6 picograms in one of the daughter cells. So it's just quite simply 6 picograms again. Then, question 5.3, stem cells are undifferentiated cells. Which statement about stem cells are true? Tick one box. Animal stem cells are found in meristems, while meristems are in plants. It's the end of where the shoot is growing, which are where meristems are found. So it's not that if it's referring to animals. Animal stem cells divide by meiosis. Well, no, that's not true because that is fertilization. Okay, meiosis. Animal stem cells would divide by mitosis, so it's not that one. Meristem cells in plants can differentiate throughout the life of a plant. It is that one, okay, because meristems, if you've got root shoots growing out of the soil, they can change into what type of cell that plant needs at that stage of its growth. Okay, the last one, meristem cells in plants can only differentiate into one type of cell. Well, that is false, okay, because they need the ability to transform into different types of cells that they need. Stem cells from human embryos can differentiate into most types of human cell. Research is being done into the use of embryonic stem cells in medical treatments. The long-term effects of using embryonic stem cells in patients are not well understood. Okay? In therapeutic cloning, human embryos are produced using a donated human egg cell and a cell from the patient. The embryo produced contains the same genetic material as the patient. Stem cells are taken from the embryo and stimulated to divide to form cells that the patient needs. The embryo is then destroyed. So effectively what is happening is they have a donated egg cell, okay, they take that genetic material out and they insert the genetic material from the patient. That embryo or that cell is then given a little shock to start it dividing, and then it can be turned into the types of cell that the patient then needs. So suggest 2 advantages of therapeutic cloning then. Well, the obvious key advantage is that if you are making these cells, they can be used to treat or cure the disease for the patient. That's the whole reason you're doing it. Okay? And also, because you are using effectively your own genetic cells, there is then a lesser chance of rejection. Okay? Then you could also have that you could have other options you could say is that it makes many types of cell or many copies of that cell, and it could also be used for research. Suggest 2 disadvantages of therapeutic cloning. Well, you are destroying a human egg. So because of that, you are potentially killing a life. So you could say, potentially, a life is being destroyed or being killed. And then another disadvantage is is obviously finding those eggs to begin with. You may have a shortage or find it hard to find an egg, or that someone is willing to donate. There is also, because this is a relatively new technology, quite a poor success rate. Okay, so we've identified 3 potential advantages and 3 potential disadvantages, but again, for each question, you only need 2. So question number 6 then is about cell structures. Draw one line from each cell structure to the type of cell where it is found. So the nucleus then is a type of eukaryotic cell. Okay, a permanent vacuole is found in plant cells, and a plasmid, that is where DNA, or how it is found in a prokaryotic cell. Prokaryotic means before nucleus. Okay, so it's a simple type of cell like a bacteria. Eukaryotic cells are like animal and plant cells. Question 6.2, figure 11 shows a plant cell. What are the names of structures A, B and C? So, and you've got this table here and you just have to tick which row is correct. So start off by taking each one at a time. So it's saying the first one, that structure A is a chloroplast. Well, A, if we label these to begin with, A is the vacuole, B is a small dot, which is the ribosome, and then part C, we're pointing to the outer part, so that would make it the cell wall. So you just need to then look at the table and which row gives you that option. Well, it's the bottom one. So vacuole, ribosome, cell wall for 1 mark. A student observed slides of onion cells underneath a microscope. The cells on the slide are not clear to see. Describe how the student could adjust the microscope to see the cells on slide A more clearly. Okay, so we can see that these slides, they're a bit blurry because they're not in focus. So what can you do to adjust that? Okay, what you could do, and this is when you've got to know the points and the names of the parts of the microscope, you've got to turn the fine focusing knob until the cells are in focus. If you just put focus it, you would actually get the mark in this question, okay? But try and throw in the names of the parts of the microscope just to make your answer a bit more scientific. Question 6.4, describe how the student could adjust the microscope to see the cells on slide B more clearly. Well, you can see B is not really magnified that much, it's very zoomed out. Okay, now we've got 2 marks on this one, so we need to add a little bit more detail. Okay, so we obviously know we need to magnify it, okay, and that would get us 1 mark. So increase the magnification, but then we would need to describe how to do this. So we should know on the microscope that our different lenses are called the objective lens. So you could say increase the magnification by changing the objective lens to a more powerful option. So that would get you 2 marks there. 1 mark for saying that you need to increase the magnification, 1 mark for saying how you do it. Question 6.5 then. The student made the necessary adjustments to get a clear image. Figure 13 shows the student's drawing of one of the cells. Figure 13, you can see that the length is 112 millimeters, okay, that they've given to you in the diagram, so you don't need to measure it yourself. The real length of the cell was 280 micrometers. Calculate the magnification of the drawing, and it's 3 marks. Now you should know the equation. Magnification is equal to the size of the image divided by the real or actual size. So we have then got, we know from our image that is 112 millimeters, and we've been told that the real length of the cell is 280 micrometers. Now we can't just divide it because these are in different units. We'll need to convert one of them to be the same as the other. Now 1 millimeter is the same as 1000 micrometers. So if I convert 112 millimeters to micrometers, that would give me 112,000. I could then divide 112,000 by 280. And if I do that on my calculator, that gives me a magnification of 400. Okay, so that is your final answer. If you didn't do a conversion and you just did 112 divided by 280, you would have got a very small number, and hopefully that should have been a prompt that you need to do an extra step in it. Okay, so if you did just leave it as 112 over 280, then that would have got you 1 mark instead of the 3. Okay, so the final question on this paper is about coronary heart disease. It says coronary heart disease, CHD, is a non-communicable disease. CHD is caused when fatty material builds up in the coronary arteries. Explain what a non-communicable disease is. Well, a non-communicable disease is one that is not caused by a pathogen. So for example, like cancer or diabetes or coronary heart disease is not caused by a bacteria or virus. Therefore, it is not spread from person to person. Figure 14 shows a coronary artery of someone with CHD. You've got the artery wall and you've got the fatty material that is blocking part of the artery. Explain how CHD can cause a heart attack. Okay, so we've got to explain, that is our command word, how this fatty material is going to cause a heart attack. We've got 3 marks, so I put 3 bullet points on my answer. What is it actually doing then? Well, the fatty material is reducing the amount of blood which can flow. Therefore, the muscle, the heart muscle in particular, is getting less oxygen. Now, if it's getting less oxygen, it's therefore not going to be able to respire effectively and therefore get enough energy. Less respiration and energy. So, and without those things, that would cause a heart attack to occur. So, question 7.3. Explain how lifestyle and medical risk factors increase the chance of getting coronary heart disease. So before we begin, I would just make sure you've got some lifestyle factors and some medical risk factors, and then you can use them to explain how they might contribute to developing coronary heart disease. So lifestyle factors that you might want to include would be smoking, obesity, and that links to lack of exercise, And then you've got obesity, eating a high-fat diet. Okay, and then you can also have things like alcohol or high salt intake. So they are all lifestyle factors that people are in control of. You've then got some medical risk factors. Okay, you might have a person that already has high cholesterol, or they might have diabetes, or they might just have in their family genetic factors that just make them more susceptible to getting coronary heart disease. And finally, they might be on a couple of medications. So what you would then have to do, once you've got these factors down, is explain how they could contribute to developing coronary heart disease. So for example, smoking gives you a higher blood pressure. Okay, obesity, again, as per the previous question, allows the build-up of fatty deposits. And then alcohol can also increase your cholesterol and blood pressure. Okay, and because you've got all of those things together, that can increase the chance of you developing coronary heart disease. So if you include loads of those different factors and then you take a moment to explain each one and how it could improve the chance of developing coronary heart disease, that would get you the 6 marks. Now, that's the end of the paper. As I've said, below are question by question links to revision videos that would help if you need to go over any specific topics, and the links to the exam paper and the mark scheme are also below as well. Thank you, and I hope this was useful.